All right, let's dive right in. Today, we're taking a journey deep inside the most complex network we know of, the communication system running inside your own head. We're going to pull back the curtain on how your brain cells actually talk to each other, a process that is behind literally every thought, feeling, and action you'll ever have. Take a look at this number, 86 billion. That's roughly how many neurons you have in your brain. To put that in perspective, that's more than 10 times the number of people on the entire planet. And every single one of those neurons can talk to thousands of others. It's a communication challenge on an absolutely astronomical scale. So yeah, that's the big question, isn't it? How on earth does this massive, tangled network coordinate anything? How does a single message get from one tiny cell to another through a sea of billions? Well, the answer lies in a structure that is both incredibly small and unbelievably elegant. So here's our game plan for today. We'll start with the brain's main connection point, then we'll look at the anatomy of that connection, what I like to call the chemical handshake. After that, we are going to walk step by step through how the message actually crosses the gap, look at the proof for how we know this really happens, and finally, we'll talk about why any of this matters to you. Okay, let's get into it. The place where all the action happens, these functional contact points between neurons, are called synapses. You can think of them as the fundamental switches and junctions for all of the brain's wiring. Now, it turns out there are two main flavors of synapse. You've got your electrical synapses, which are kind of like a hardwired connection. They're super fast, but simple. But the ones we're focusing on, and the vast majority in your brain, are chemical synapses. They use these little chemical messengers to send the signal, which makes them a bit slower, but way, way more versatile. And that adaptability is the absolute key to things like learning and memory. So let's zoom in and really look at the anatomy of this chemical connection. Before we can understand how the message gets sent, we kind of need to meet the key players involved in this microscopic handshake. And this image lays it all out beautifully. On one side, you have the presynaptic terminal. That's our sender neuron. And you can see it's packed with those little bubbles full of chemical messengers. On the other side, you've got the postsynaptic neuron. That's our receiver. And right there in between them is that tiny, tiny gap. That gap is called the synaptic cleft. And this is so important, it's a physical space. An electrical signal can't just jump across it. This gap is the entire problem that chemical signaling evolved to solve. So the question is, how does the message make that leap? And that brings us to the absolute heart of this whole thing. We're now going to walk through the elegant, step-by-step process that allows a signal to travel from one neuron to the next. So here it is, the blueprint for how neurons talk. Now, I know it looks a little complicated at first glance, but trust me, we're going to break it all down step by step, and it's all going to make perfect sense. Okay, so it all kicks off with an electrical spike, an action potential, that zips down the neuron. When the signal arrives at the presynaptic terminal, it's like a key turning in a lock. It flips a switch that causes these special little channels to swing open. And that opening allows calcium ions to just flood into the terminal. Think of it as the signal arriving and just opening the floodgates. This sudden rush of calcium is the crucial trigger. It's the signal for all those little bubbles we saw, the synaptic vesicles, to travel to the edge of the neuron and fuse with its membrane. This process releases their cargo of chemical messengers, the neurotransmitters, out into the synaptic cleft. They then just drift across that tiny gap and bind to specific receptors on the other side, kind of like a key fitting perfectly into a lock. So once those neurotransmitters lock in, it causes new channels on the receiving neuron to either open up or close down. This changes the flow of ions, which creates a whole new electrical current in that cell. And this new current will either excite the next neuron, making it more likely to fire, or inhibit it, telling it to quiet down. Either way, the message has been delivered. But the job isn't done yet. To get ready for the very next signal, that gap needs to be cleared out. So the neurotransmitters are quickly removed, either getting broken down by enzymes or basically vacuumed up by other cells. And at the same time, the neuron recycles the leftover membrane from the used vesicles, pulling it back inside to be refilled for next time. It's an unbelievably efficient system of communication and recycling all in one. Okay, so that's a really beautiful model, right? But how in the world do scientists know that's what's actually going on? Well, let's take a look at the clever experiments that gave us the proof. A huge piece of the puzzle came from Bernard Katz. He figured out that these neurotransmitters aren't just released in some continuous stream. Instead, they're let go in these standardized little packages, or quanta, as he called them. Each packet contains a very specific amount of the chemical messenger. And a lot of this foundational work was done right here at what's called the neuromuscular junction. This is where your motor neurons connect to your muscle cells. And the reason they studied it is because these synapses are huge and pretty easy to access, which made them the perfect place to watch this whole process unfold. And using powerful electron microscopes, scientists could actually see the physical source of these packets. They're those synaptic vesicles we keep talking about, each one loaded with thousands of neurotransmitter molecules. So the theory was simple. One vesicle equals one quantum, but they still had to prove it. This is the crucial part. Researchers John Heuser and Tom Rees came up with this brilliant experiment where they could stimulate a neuron and then instantly flash freeze it, stopping everything in its tracks. Then they could literally count how many vesicles had fused with the membrane. And when they compared that count to the electrical signal that was measured, they found a near-perfect one-to-one match. One vesicle fused, one quantum of signal was released. That was the definitive proof, the smoking gun. So, after going through all of that incredible detail, from calcium ions to recycled vesicles, let's zoom all the way back out. Why does this tiny, lightning-fast dance happening inside your brain matter so much? Because this is it. This microscopic process is the biological foundation for everything that makes you, well, you. Every thought you have, every memory you form, every emotion you feel, and every single action you take is the end result of trillions of these chemical handshakes happening every single second. And that leaves us with one final big thought to ponder. If this impossibly intricate microscopic dance is what defines our reality, it really makes you ask, what happens when the music changes? What happens when the timing gets off, or the wrong messengers get sent? You see, understanding this dance isn't just about understanding the brain. It's about understanding the very foundation of ourselves.